Hello, good afternoon. Yeah, we have a few seconds more for the presentation to start. Less than 7 seconds. Please comment, say hi in the comment section. Good evening, everyone. This is another opportunity for us to be able to meet this evening again. And this time around, we are coming on a good ground because it is what is expected of us to be doing in the next few months. So today we are going to see what are actually expected, what should be the project guidelines for us as a microbiologist as well as a bioinformatician. For some of us that presented today, you will see that you are asked to pick randomly a number between 1 and 4. And just like it is being highlighted here, you will see that cohort 1, that is subgroup. See, don't forget that this group of my project student for 2025 is being divided into four. Group one, which we call cohort, is those that will be working in the laboratory. So, and that is what is called the wet lab. Wet lab. It has its own advantages and it also has its own disadvantages as well. But don't forget that whatever is going to be a laboratory, those that will be in the laboratory will have very limited resources when it comes to the issue of laboratory based. So we can only move up to the level, uh, primary level, that we will need it to take our sample outside for analysis. So that we can only work at morphological, up to morphological and physiological aspects before that. Then the cohorts 2, 3, 4, these are the cohort group that will involve the use of bioinformatics. So don't forget, some of us we are not new. I'm very sure all of us, we are not new when it comes to the use of bioinformatics. Then some of us, we don't even know one of the reasons why bioinformatics is very, very important in the designing of our project, use of bioinformatics in our project. Just like I said, cohort 2, that group is going to be group for vaccine design and development, and group 3, that is going to be for drug design cohort group, and group 4 is going to be for AMR group, that is antimicrobial group. And don't forget bioinformatics. What do we mean by bioinformatics? We are not new into defining bioinformatics, but we are so new in using bioinformatics as our own final project. Bioinformatics is the use of computer, that is computer based analysis. So I told you that group one, which is laboratory based, is wet lab. Then group two, three, four, this group comprises of what is called the dry lab, because you don't, where you are going to work is not going to be a wet lab. You don't need to mix one reagent with another one. Once you have your source of internet and you have your computer, honestly speaking, you are already working in bioinformatics cohort group. And just like I said, so today, what are those guidelines? All these cohort group 1 to 4, what are the specific guidelines that you must follow? That is why we are here today. Okay. The objective of this project, number one, is to understand the essential methodologies in microbiology. What are the methodologies? What are the methods? What are those materials that we will need in order to be a cohort group, in order to be in any of these cohort group, be it wet lab or is it going to be in a dry lab. Another name you can give to bioinformatics group apart from computer um based group is what we call in silico. In silico means in with the use of computer in silico. Okay, so just like I said, you can broadly divide my cohort group into laboratory based, that is a wet lab, and bioinformatics. So that is dry lab. And those in bioinformatics group, just like I said, comprises of vaccine design, drug design, and antimicrobial resistance. Okay, now we are starting with what are the guidelines. People that will be in the laboratory, what are they going to do? So introduction to laboratory based project, that is the wet lab. The focus is going to be on identification. You are going to identify and isolate microorganisms using their using morphological techniques. Don't forget that microorganisms, they are in their natural state. For example, you find if you are working with a soil sample, that means that is their natural habitat. So you need to take them into the laboratory and be able to culture them, be able to isolate them. You should be able to grow them in the laboratory. And that's why we have to talk about the use of media, nutrients agar, no, sorry, nutrient media, the use of agar. Agar is just a kind of laboratory based habitat. So you are going to imitate all the condition that they have in their natural environment. And that's why laboratory based project is going to involve identification and isolation. And under identification, we have colonial identification, that the use of how they grow in the colony. When you culture them, you introduce them into petri dish. How do they grow? So we will see it. Then we can also use their morphological appearance under the microscope. Gram stain under the microscope. Do they move? That is motility testing under the microscope. Then we will also do a little bit of biochemical test, that is based on the enzyme that they contain, based on the enzyme that they are made of, are they able to metabolize hydrogen peroxide. So we will see this, and that is called the oxidase test. We have oxidase test, we have catalase test. Hydrogen peroxide is the catalase test. Catalase, that is, do they possess uh, the enzyme that can convert hydrogen peroxide into oxygen. So we will see that. So that is part of identification. For identification, we go on to the lowest level, which are the molecular analysis. That is why whatever identification we have to do, we have to make sure that you identify to certain level. The other level is when you are going to export your work for PCR. When you extract the DNA, you do the DNA extraction. That is, you have to know them. And don't forget, in your elementary microbiology experience or elementary biology, don't forget that microorganism, dey possess their own DNA. And if you, until you get to the lowest level, which is the DNA, before you be able to say that is what the organism that you can do proper identification. Sometimes in the laboratory, there are different types of a microorganism that they will have similar enzymes, and at the end of the day you are going to mischaracterize them, you are going to misidentify them, thinking that they are a specific type of microorganism, whereas at molecular level it might not be so. So that is why. So the summary of what is called the laboratory base should be isolation and identification. And this identification will start from their colonial appearance up to the last level, which is sequencing, which is partial sequencing, that is molecular analysis. So that is cohort one. Now cohort group, yes, still under cohort group, I have explained this. That is we use agar plates. Agar plates, that is petri dish, different types of selective media. We are going to see this, and different types of environmental condition. Environmental condition include, there are some microorganisms that they will grow at certain pH, they will grow at acidic pH. There are some of them that will go at normal atmospheric temperature. So, and there are some of them that will not even be able, there are some that will grow under aerobic condition, and there are some of them that will not even grow under aerobic condition, anaerobic and aerobic. So all these are part of what we are going to explore when we talk about culture based, which is laboratory based isolation. So just like I said, there are four key steps in isolation. Number one is you have to remove them from their natural environment. For example, if you are working with a soil microorganism, so that means your sample, the sample you are going to collect is going to be what? Soil sample. So you cannot say that you are interested in soil microorganism, and you go and collect blood sample. It is not done. So the first step is, how can you collect the normal that can give you the necessary microorganism that you are actually looking for. So that is telling you that the first thing for you to be able to do is, can you collect the correct sample, correct sample. So sample collection. Then another thing is preparation of media. Preparation of the media is just like when you are preparing food for the microorganism. You want to prepare food that will make them to be able to grow as if they are still in their natural environment. So your skill in in preparation of media should be very, very, very good. So that is preparation of media. Another one is inoculation of your sample. After you must have prepared your media, the next thing is to introduce that sample into the media you have prepared. If you have a very good sample collection, you have the correct collection. Instead of you to collect soil sample, you collected soil sample. And apart from that, you also prepare the food, delicious meal for them to be able to grow well. But after preparation of dis delicious media, you don't know how to introduce them correctly. So instead of you to introduce them in the normal correct procedure, or correct procedure, you are using a different, which is inadequate. So you are not able to introduce them correctly. So you just think you have some of us just, by the time you use your wire loop, there are some of them that you can use wire loop to introduce, whereas there are some that you need syringe to introduce. For example, any sample that is going to be in form of a solid, so I think you need to first of all do what is called a serial dilution. Liquid, you need to do serial dilution. There are some that by the time you want to introduce them, you do serial dilution before you inoculate them, you introduce them into that. And there are some of them that you don't even need that. So the next one is incubation and observation. After you must have inoculated them, so the next thing is you should be able to leave them in the correct, you leave them inside the incubator, and wait for between, if it is bacteria, you wait between 18 and 24 hours. you will see them already growing because they already have uh, good food, appropriate food for them to be able to thrive well. So if you are working with fungi, that one is going to take you a longer period. That one can take up to 5 to 7 days before you'll be able to see them growing. And if you are working with something like yeast, is because that yeast is also a fungi, it's going to take a longer period. And there are some of them that you might not even be able to even see. For example, if you are working with Mycobacterium tuberculosis, that you have to wait up to 8 weeks before you see them growing. But whichever one, after you must have introduced them into that level, you still need to do what? Incubate them, then you will be able to start observing them. So different types of media that we will use for this job, for this work. Those of you that will work in the laboratory will need the following. The first one is a general purpose agar. General purpose agar, an example of that is a nutrient agar. That is what we are going to make use of. We call it general purpose because it is not only bacteria that can grow on them. It is just like all of them, just as if in Yoruba, everybody must have a source, at least a source of carbohydrates. So then we also need a selective. So example of broad-based agar is nutrient agar. The next one is selective media, which is McConkey. McConkey agar is part of a selective agar. So then we will also make use of enriched agar or enriched media. Example is that of chocolate. We are going to see the way to prepare chocolate and the way to prepare blood agar. It is already enriched with blood. That's why they call them enriched media or enrichment agar. So example is chocolate and blood. Then differential. In this, we are going to make use of McConkey and SSA. And for example, if you want to buy general purpose agar like nutrient agar, each of them will cost minimum of 70,000 naira per 500 gram, and that is what we will need. Okay, they are going to. Then, what are the morphological characteristics that you can use for identification at this level? At those that will be working in the laboratory, they need something characteristics like their size. What is their size? You have to pay attention to the shape, you pay attention to the color, you pay attention to the texture of, is it mucoid, is it slimy, yeah. So you have to texture of this agar. So then understanding the basis of gram stain. Don't forget that they can either be classified into gram positive or gram positive or gram negative. Those one that based on the composition of their cell wall. This is not the first time you'll be hearing that. Based on the composition of their cell wall, that one will classify them into either gram positive or gram negative. Although there are some of them which might not be our focus in this work. So then there are some of them you identify through their motility, motility testing. And there are some of them that you have to remember your STM325 that has to do with metabolism. Metabolism, the ability to metabolize glucose into pyruvic acid or into CO2 and so on. You have to know that. Then the one that is going to be above us here is a molecular identification, which has to do with we have to extract their DNA first, then we make use of a polymerase chain reaction, that is we amplify that DNA, that small fragment of that DNA, we amplify it for specific gene. Then after that, we now sequence the whole gene so that we'll be able to know the order. Is it going to be CAT, CCC? That is the order, wanting us to know what is going to be. So then after that, after we must have gotten this, I told some of us that even when you must have gotten your results of sequencing, 16S ribosomal RNA sequencing, that is partial sequencing, You still have to subject it to bioinformatics analysis. So then, tools, what are those tools that we will need? Are going to need PPE, that is personal protective equipment, like your lab coats, like your hand glove, like your nose mask, everything that can protect you. That's why they call it PPE, personal protective equipment. You cannot be working in the wet lab without putting on this. If you are working on bioinformatics, you don't need PPE. What you need is just your computer. So it's a dry lab. So one of the disadvantages of wet lab is that is the this level of contamination. You are you are more you are more exposed to contamination, sorry, to get contacted with all the, because you will be working with a live organism that can cause infection. So you are more exposed to that. So one way or the other, you can contact any of dis microorganism. If you are not so careful, then you need lab notebook. Everybody, you must buy hard notes, that is hard cover, for you to be able to document. Every day you should be able to document. And don't forget that as I said, I told some of you that you need to prepare ahead. Before you get to the laboratory, you must have written out the procedure you are going to use, and the protocol, disinfectant, that towel, all this one. This is these are part of the consumables that you will need. Then I told you that you have to remind me, so that we have to put at least one binocular microscope in the department into proper working condition. So, and we also need something to power it, that is solar generator for its usage. So remind me of this, so that you should be able to make use of microscope effectively. If you are a microbiologist or a biologist and you don't know how to handle, you don't know how to do microscopy, that is not too good for you. So this work is going to help you to do that. Then don't forget your data collection and documentation. Documentation is very very important. Right away from when you are collecting your data, you start documenting, you start documentation. So what you should do is just every point, every time, you should be documenting. If you are not documenting, if you are not writing on anything down, honestly speaking, you will forget a lot of things, and you will find it very difficult to, even when you are getting results, you won't be able to remember the procedure that you have followed. And don't forget that that is very very important in record keeping. Is part of the backbone of being a scientist. Recording colony characteristics and experimental condition. So you have to be documenting them. At what time, including the time of collection, the time you are doing that, all the procedure, detail of your procedure. That's why your laboratory note is very very important, and your documentation has to be daily, daily. So don't say that today you have done isolation or just sample collection, and you won't be able to document the second day. Every time, every day, you have to use documentation for that. And ChatGPT should be your AI assistant. I've told you that it should be your closest companion. For example, if you want to do gram staining, what are the procedures? Consult ChatGPT. If you want to do biochemical test, what are the basic biochemical tests that you need to do? So you have to consult it from time to time. So ChatGPT should be your assistant. So, and laboratory safety protocol, just like I said, is very very important. Now for those that will be in cohort group 2, that is vaccine design group, that is vaccine designer. Number one, the first thing is that you have to understand what bioinformatics is. Bioinformatics, if somebody, you, at the end of the day, I saw some people that they use bioinformatics as their own project. Drug design, vaccine design, and they just ask them that simple question, the simple question of what do you understand by bioinformatics. They don't know. They could not give any correct response. It is very very shameful. So how can you use bioinformatics as a tool for your project writing or for your project execution without knowing what bioinformatics is all about? So you have to know bioinformatics, definition of bioinformatics, application of bioinformatics, how to retrieve data, what are the databases that you have. So you need to know, and its application in vaccine design. So apart from bioinformatics, you need to also have good knowledge of what vaccine is, different types of vaccine, which one are you going to produce. You should be able to also understand the biology of the organism you are interested in. We will see it as we go on. Then the computational method in designing vaccine. What are the advantages of using computer base, of using bioinformatics for you to be able to design your vaccine, apart from traditional way of designing vaccine can last you for more than 20 years before you get a vaccine. But when you are using computational method like bioinformatics, it can, in fact, within 1 year you can even get vaccine for it. For example, COVID-19 came in 2019, and by 2020 we already have vaccine already that will be used for what? For to control COVID-19. So that is very very important. So understanding pathogen biology under normal circumstance, you need to pick up. Do you want to work on bacteria, or you want to work on viruses, or you want to design? So you need to pick any pathogen, that is disease causing organism. Which one are you going to pick so as to design vaccine for? So whichever one you now pick, you must know the biology of it. You should know the mode of transmission, look at the molecular biology of it, look at everything about that organism, look at how notorious are they, look at it. So that's why we said overview of pathogen genomics, e.g. sequencing the genome of bacteria and viruses. So the first thing is you have to identify the potential antigen. What weapon do they have? What weapon do they have that they are using as a antigen? Don't forget that I told you that antigen, they are foreign. Apart from being foreign, they have the ability and capability to be able to trigger an immune response. So, and what does vaccine does? Vaccine train our immune system in order to be able to fight against infection. Dey train our immune system, dey prepare dem in case of war. That is vaccine. So you should be able to understand what are the characteristics of vaccine, what are the attributes, what are the properties of this vaccine. So you suppose to know that. And each pathogen has its own unique antigen which must be specific. For example, the antigen you see in viruses are quite different from the antigen that you see in bacteria, and it also varies from species to species. It also varies from one environment to another. It also varies from one condition to another. For example, when the same E. coli that you isolated from soil is going to have a different antigen apart from the one that is isolated in water. So you have to know all this. Each pathogen has its own unique weapon, antigen, which are specific. Then data collection, you need, where are you going to get all this? You need to, I told you bioinformatics, you get some of these antigen in form of amino acid. You get them from the following databases. Uniprot, you are not new to Uniprot, you are not new to PDB, that is protein database, NCBI for pathogen genome, and we are working with tuberculosis, that is tuberculosis. there are some bacteria and viruses that they have their own specific website or database for that. So that means you need to know. Importantly, understanding the pathogen biology and molecular structure is very, very important. Then after you must have gotten your data from all these databases, the next thing is that you should be able to retrieve of your data. You have to clean your data, clean them, so that this. Then you have to know what is called a sequence alignment by the use of BLAST. This should not be the first time you'll be hearing BLAST. That is a basic local alignment sequence tool to compare different pathogens and their DNA sequences. So then you have to avoid antigen. You avoid antigen or antigenic protein that can form a kind of cross reaction with human antigen. So if the antigen you are getting, you are retrieving from that database, if you subject it to, if you do alignment and you discover that they form a kind of similarity with that of human, that means it's not going to be a good vaccine, because a vaccine should not be able, is not part of human body, but it should be able to serve as a foreign, so trigger an immune response. So epitope prediction, so we need the following databases as for us to be able to predict epitope. Epitope, IEDB, Bepiprid for predicting epitope, selection of target epitope for vaccine. Then the next question for you is what do you understand by epitope and how important are they in vaccine development? So you need to know all this. Then at a point in time, you also need to do what is called modeling protein structure using tools like Pymol, Chimera for three-dimensional modeling of antigen. You are going to see all this, and this is part of what you will see by the time you go on. Then antigen refinement and validation, you also need to do that. Then immunogenicity prediction, you have to predict their ability to be able to trigger an immune response. If at the end of the day, the antigen you have retrieved, they are not immunogenic, that means they won't be able to serve as a vaccine. Don't forget that vaccine, they are going to be, they are just like an antigen. They should be foreign, and they should have the ability and capability to trigger an immune response. We are going to see more of this as we go on. Then finalizing vaccine candidates, that will be final validation using some in silico model before we do what is called laboratory testing. So an example of where they use bioinformatics to be able to design this is the mRNA vaccine like Moderna and so on. So clinical trials is the vaccine you have designed. You now gather um, laboratory animal like rats, and you test it on them. So we don't need that because this. Then the cohort 3, that is bioinformatics, which is for drug design, using bioinformatics for drug discovery. The first step is target and identification, that is protein receptors and their enzyme. So that's the first thing. The next one is a drug target interaction, which is very, very important. Importance of studying interaction between drug and target protein. You need to learn what is called molecular docking. So we will do that. We will be able to do that by the time those that will be in that group will need to do that. And for some of us that will make use of the past, just these past project students, my past project student copy. So you'll be able to also follow that. Then virtual screening, you have to screen the use of computer. You screen whether all categories of a compound, you can screen up to 150. You screen as many as possible. Would they be able to be used as drug? So that is facial screening. Then the software we are going to use include Autodoc, then Glide for facial screening of compound. All these are part of what we will use for that. Then the drug databases include PubChem, Drug Bank, Chemebil, key resources for this. Then screening of compound laboratory. Okay, I told you molecular docking. we need you need to know all this by the time we go on. Don't be afraid. All this, you as time goes on, you will also know and learn how to make use of them. But don't forget that you need to read more in order to be able to do this. So there are some of them that I will be able to put you through, and there are some of them that you need to also consult ChatGPT, consult people that have actually worked on it. I remember that this past year, last year was the first time I had students working on drug design, and they were able to do it successfully. uh, Prominent among those that work in that are Aishat and Zainab, two of them I know. So even if you are stuck and you don't know where to go, you can have their write up as well that you follow their step. Then you can also contact them in case you have any problem at all. Then we talk about QSAR modeling. All these are part of what we do for drug refining. So you still have to refine them. You need to get them in pure form, in their pure format, not so then admet prediction, that is uh, this is to measure their toxicity, absorption, distribution, metabolism. This is what is called the toxicokinetics of that drug. We need to know how, if they absorb, we need to know their absorption level, distribution level, metabolism level, how will they be excreted. Then we need to also know, are they going to be toxic or are they not going to be toxic. All these are part of the molecular tools that we also make use of uh, our own computer to be able to predict all this. So don't worry that maybe you need to go and study computer. No, you don't need any other computer. This, you get used to them by the time you start working on them. Then drug optimization, that's another step on drug design. Optimizing the drug molecule for potency, selectivity and minimal side effect. This is very very important. Then normally there should be a preclinical and clinical testing, but we are not going to do that. So ethical consideration in drug design, that is regulatory bodies and ethical standard in drug design, that is federal drug agency and so on. So we don't need all the food and drug. So cohort group 4, that is bioinformatics, and what that is called AMR group. So what do we understand by AMR group? The full meaning of AMR is antimicrobial resistance. Antimicrobial resistance. So we are also going to make use of a computer to be able to use antibiotic to study antibiotic resistance in microorganism. Okay, so part of the databases we are going to use, this just two out of it, is not only limited to this. We use cards, comprehensive antibiotic resistance. In fact, we make use of this. Then there are newer ones like res finders. We make use of these databases and some other databases that we can use. Then genome sequencing for antimicrobial resistance. So we use the first step is using sequencing technique to be able to analyze resistant strains of bacteria. We will make use of all this by the time we get to that level. Identifying resistant gene through genome sequencing. So all these are very very important. Then understanding resistance mechanism. So under normal circumstance, how do resistance develop in each microorganism or in each bacteria? How do they develop? What are their mechanism? Is it through is it through efflux? Is it through inhibition and so on and so forth. So mechanism include enzyme production. There are some of them that will also produce some enzymes, like the enzyme they produce to render penicillin inactive include beta lactamases. So there are some of them that they will membrane permeability. They will make their membrane to be permeable to that drug. So they will just change the permeability level. if it has been easy for the drug to enter before, they will now develop additional codes to make that drug not easy to enter into this cytoplasm. So identifying resistance chain, so how to analyze sequencing and so on like that. Okay, so resistance gene mapping using of bioinformatics tool to map resistant gene. will Then predicting cross resistance. There are some drugs, there are some antibiotics that all bacteria or all microorganisms can develop resistance to. You use, if you for example, isolate ABC, you can see some of them can develop resistance to penicillin all through. So that is what is called cross. So we will also make use of a computer to be able to predict that, so that which of these drugs can they develop cross resistance to in the future. Don't forget, by the time you are trying to study the antimicrobial resistance, we are indirectly trying to predict the resistance that will be coming in the future, so that even if you want to to develop any drug, you'll be able to predict even beforehand whether that drug is going to serve the purpose or not. But don't forget that antimicrobial resistance is a global threat to public health. There are some drugs that were so effective in some years ago, but now they are no longer effective because microorganisms have actually developed resistance to them. So by the time you are trying to predict antimicrobial resistance, you are indirectly trying to advise the WHO, you are trying to advise people, you are trying to advise uh, pharmaceutical companies of which drug and which line they should be able to follow. Okay, case study like antimicrobial resistance in tuberculosis. The real world example now is that you now have mycobacterium tuberculosis that are what? They are total resistant to certain antibiotics. Okay, that is no antibiotic that can be used in even curing them. Okay, so this we make use of um computational techniques in order to be able to predict that. So then apart from that, we have to know how to read heat map, resistance map and cluster analysis. These are part of what we will need for AMR data visualization. Then data analysis and interpretation is very very important. Analyzing experimental data for all cohorts, statistical method, graphs and tables and so on. So writing scientific report, after all this, we will be able to know how you can write all this together, write it in form of abstract, introduction, methodology, results, discussion, conclusion, recommendation and your references. So um then now, what are the common mistakes to avoid? Mislabeling your data. Mislabeling your data. For example, instead of you to label your data, you need to label your data correctly, you are labeling it another one. Then poor experimental control. Some of us, we don't even know what experimental controls are. So that's why I want you to answer this question and you should send them to me. What are experimental controls? Explain what do you mean by negative and positive experimental control. So then organizing your project timeline. So you don't have all the time. You have to set your time, you have to set your milestones. What are you supposed to get at different time? You have to set there. So how can you collaborate effectively in your group? So you should be able to, you should not work alone. You are not Liverpool. You should not work alone. Even Liverpool can never work alone. So what we are saying in essence is if you want to work and you want to be so effective in your project work, there is nothing like individual work. That you are going to work alone. No, it's not possible. So you have to collaborate, interact with others. You bring out ideas, you share ideas, you work in group, work in pairs. So for you to be able to do. Then overcoming challenges. How can you troubleshoot common issues during experiment or bioinformatics analysis? Challenges will surely come. The road is not going to be smooth. the road is going to be very, very rough. So many times you feel like crying, you feel, how will you overcome all this? It's part of what you should do. So how can you do that? So then the final tips of, so stay organized, communicate effectively, and keep a constant workplace. Okay, then I just put here a rough budget of what and what will be needed, at least if you are going to work on this. For example, consumables. I told you part of consumables are those media that you need to buy, and I've also mentioned like 5 or 8 media. So, and each of these media will sell nothing less than 70,000. So if you now have 8, that is 70 times 5 already. That's already more than 400,000. So reagent are staying that you will need, some equipment that you will need. I said we will have to put a microscope and some other things. We have to put them in proper shape. Other things that we need, then molecular identification. After all this is just a rough estimate, and it's not only for everybody. It's just a kind of rough estimate. This is the minimum per what you need per person. And now if you want to now do per isolate, if you now want to do molecular identification, you need nothing less than this one. Okay, and as I told you that you need minimum of 17 to 25 weeks if you are working on a laboratory based project. That is that. Then propose budget for cohort group B, that is for fashion
How is the
design,
boiler?
is what you have, like software licensing. We are going to buy a lot. I mean, there are some licenses that you have to buy. Okay. Then apart from that, data access fee, computational resources, then the source of internet where we have to subscribe to the business plan, styling, which is around 160 per month, and so on. At least rough estimate is just a total of that. Then the second group, which is fashion design, which is 16 to 25. Okay, so it's going to 16 to 25 weeks. So that means from now, from the first time you start, you need between 16, minimum of 4 months, and maximum of 7 months for that. So that is telling you that the best time to start is for you to be able to start your work right now. Start it early. The earlier you start, the early you will come out of all this. Okay, then drug design, you also need something like this apart from other procurement that you need to make, apart from other procurement that we will make in that cohort group. Then they also need between 16 and 24 weeks. Okay, that is 4 months to 8 months. Okay, for that work. Then for antimicrobial resistance cohort group, these are rough estimates, and that's the minimum. That's the minimum. It ranges from 300,000 to, even you can even spend as much as 3 million per cohort group. So it depends. Okay, then that one also need to, from the first time you will start, the first phase is going to be 3 weeks, minimum of 3 weeks, followed by 4 weeks, followed by, all these are just rough estimates. You can spend less and you can spend even more if care is not taken. Okay, and cross resistance visualization, that's going to be for 3 weeks. Data interpretation and reporting, that's another 3 weeks, making a total of 16 to 24 weeks. Don't forget that honestly speaking, we really have to start. When is the best time to start? The best time is to start now. The best time is never to delay again. The best time is once you have gotten, you know, the group you have, you belong to, the best time to start is now. Some of you that already you don't know the group you are going to be, by the time you come for the presentation in the next few, this same week will not pass that you will know the group that you belong to. So after knowing the group you belong to, we now start talking about the topics we are going to focus on. I will give you assignment, I will give you task, go and get where you want to work, what microorganism you want to actually work in, the topic before you begin, you continue. And don't forget that the target is that by March next year 2026 you should be able to be at the final stage. Den don't forget dat you need to do proposal writing, which is pre-data proposal writing. You have to write proposal on whichever, so dat is telling you dat whatever topic you are going to pick on, whatever whatever topic you are going to work on, you should talk about what? About the pre-data. You will do the data presentation. Then I think February, is it February or next February next year? That is you are expected February next year you are expected to do your post analysis. That is when you are supposed to go for external. Your external examiner must have come and examine your work. So you just have between this October and you have just 4 months to be able to work. So if you really want to graduate, I'm very very sure you don't have more than that. So that's why I said it is better you start early. It's better you start now. If there's any other time you need to start, it is now, not any other time. So don't forget, if you are still on the line, if you are still with me, please don't forget, make comments. Say hi in the comment section. Then don't forget that I gave you an assignment, and that assignment must be submitted in the next 1 hour. Must be submitted in the next 1 hour. So when you flip through and you listen to it again, you are going to see where I gave you assignment and what is actually needed for you to be able to do. Once again, I really want to appreciate you. Once again, this is a comprehensive guide for lab based work as well as bioinformatics. So then I said that generally speaking, I've divided my own group into 4 groups. The first one is wet lab. That is the laboratory based microbiology students, laboratory based students. Some in environmental biology will still join them. So don't be confused that because I am emphasizing on microbiology. No. So so far, but the work they will do is still going to be tailored in that area too. Then the bioinformatics group are the one that we have them as a dry lab. They are not going to add any reagent together. They are not going to use um any consumable. So what they will just be using is their source of internet and their computer to do whatever they want to do. And don't forget that everybody, whether you are working in the lab, whether you are working using your computer, you must know that biology, that bioinformatics is nothing but the use of computer to be able to solve biological problem, to be able to solve or design biological problem. And I said there are 3 different groups: vaccine group, antimicrobial resistance, as well as drug design. So all these are part of what you should know as a take home in this presentation today. So please, if you are hearing me once again, please say hi if you are still hearing me. If you are not hearing me, say nay. If you are not hearing me, say nay. If you are not, if you are not hearing me, say nay. Okay. Ah, once again, I want to thank you very well. Thank you very much because of for your time today. So I want to believe that the next time we will do. we'll be able to do more in this. So before the week runs out, maybe or either on Friday or later on, we'll still talk about another thing on our project design. So once again, I really want to thank you. So